Today, what we're going to be working on, well, actually, you're obviously on some task somewhere in the Sci-Hub process. What I'm going to do today, though, is I want to introduce task number 5 to you. If you're not on task 5 yet, then you'll be doing task 5 tomorrow. Because what I really need to do is I need to explain some of the most important aspects of how we can successfully do this assignment, the experiment. And you'll have just enough time to start and finish task 5 today if you're starting that. If you're one of those people that maybe you're still finishing up task 4, that's an okay place to be in. You'll just do task 5 tomorrow. There's no problem with that at all. Regardless, this is just a tough experiment to fit into one class time, but it has to happen in one class time. You can't split it into 2, so we have to get it started and finished today. So what I'm going to do right now is I'm going to go through the steps of kind of some stuff you need to know before you can start the experiment. But the experiment has a long list of instructions of what to do. As you go through that long list of instructions, though, I'm going to give you some information that will hopefully make it more understandable as to how you're going to go through those instructions. Okay. So to begin with, let's have a discussion about our experiment by answering some of the questions that I have up here on the board. So if we look at what we're going to do right now is we're going to work on proving a pattern. Okay, we need to prove a pattern about the carbon cycle. All right. So my first question up here says, why do people say that we need to plant more trees when we talk about carbon, the carbon cycle? What are trees going to do for us? Why would we want to plant more of these? Abby?
They're going to do photosynthesis. You don't have to, but they're going to do that.
Photosynthesis. Go ahead, try that. Say that for me.
Photosynthesis.
Okay.
It feels weird. I don't have my excited in my mouth anymore. It just.
Oh, nice. So the roof of your mouth is gone, huh?
Yes.
Yeah, I know how that goes. I remember that.
And the plants take the carbon dioxide out of the air with water, sun, and sunlight, and then they create it. And the plants create it into carbohydrates for us to breathe in, along with oxygen.
Okay, so the reason we want to plant more trees, if I could summarize what Abby was saying in a simple, just one sentence, I'd say plants remove carbon dioxide from the atmosphere. That's why they say plant more trees, because the more trees we have, the more carbon dioxide that's sucked out of the atmosphere by those trees. Now, also, like you said, though, Abby, trees also produce oxygen, which is good for us. We don't need any more oxygen, but it's better. It's a good waste product. Plants poop out oxygen. You can think of it like that because it's their waste. We don't necessarily need more oxygen, but their waste product is a good thing for us. It's not a bad thing, the way that they create waste. They're making oxygen. So it's okay to have trees. If we had something else like our waste product, when we breathe out carbon dioxide. So if we say plant more trees, this is good because it removes CO2. Now, when you guys were doing task 4, you were looking at carbon stores, where we store carbon, which is called a carbon sink, or a producer of carbon. So are plants considered a carbon sink or a producer of carbon? Okay, so this is going to be a carbon sink. So if we remove CO2, think about this. If you pour something into the sink, you get rid of it. So this is a carbon sink. So we need to now prove the pattern. We need to prove that trees are truly going to remove CO2. All right, the way that we're going to do this is this experiment that I've called the carbon capture technology, because plants literally can capture carbon out of our air. So down here on task 5, carbon capture technology, you'll use this button to get to the instructions. So in this experiment, we're going to use spinach leaves to prove that plants convert carbon dioxide into oxygen. So we're going to prove that that is truly going to happen. In order to do that, there's several steps here, and then there's some experiment procedures here. Now, throughout the day today, so far, I've noticed that you need every moment of class today to do this procedure. So if you're on task 5, go ahead and skip this where it says to complete this slide. You can do that tomorrow. That'd be a good thing for you to do while you're in class tomorrow. Today, go ahead and get the slides, and then just start number 3, where you'll start the procedure. So to give you a little overview of why there's 2 procedures here, what's going to happen is we need to do 3 parts in this experiment. You're going to prepare the solution, then you're going to prepare the leaf pieces, and then you're going to do the experiment. So when you prepare the solution, there's two different solutions that need to be prepared. So you're going to get two groups. Let's say we have group A and group B that are going to work together. So you can have one person in group A and one person in group B, and you have a pair of two people that are going to work together. Or you could have two people in group A and two people in group B, and you can work separately, and then you work together as a group of 4. You can even have 3 in group A and 3 in group B, and then at the end you come together and have a group of 6. That's okay. So the reason that we need to have two groups, though, is because group A is going to prepare a solution, and group B is going to prepare a solution. And when I say a solution, what I mean is we're going to have beakers that we're going to put water in. One of them is only going to have water in it. The other one's going to have water and CO2. Now, the reason is because we want to make sure that CO2 is the only thing that's different between these two beakers. So what do we call the one that doesn't have the CO2 in it? To prove that CO2 is the thing, we have to run this one over here without CO2 to prove that it's not going to work. Does anyone remember from like August, what is it called in an experiment when you run 2 trials? One of the trials you test your thing, the other trial you leave it normal to prove that the thing you test is truly the reason that it's going to happen. Remember, Andrew?
The control variable.
Okay, so this is our control experiment, meaning that we're going to control this to make sure there's nothing that's going to be added. Over here, this is our test one, meaning that we're going to test to see if CO2 is truly the reason that plants go through photosynthesis. Okay. Now, what you'll do once you set these two up is you're going to do several steps to get all of this all prepared. At the end of those steps, it's going to tell you to come together as a group. And when you put these two beakers together and you come together as a group, that's when you'll do the experiment, and you'll put both of those beakers next to each other. So you have beaker A and beaker B next to each other. So that's why we start as 2 groups, and at the end you come together and put both beakers under the same lamp, because one of the beakers is going to have CO2 in it and the other one won't. All right, so what we need to do is we need to understand what Alka-Seltzer is, because I'm going to give you some Alka-Seltzer to allow this experiment to happen. Okay, Alka-Seltzer. On the container here, it says that it's an antacid. Show this to the people at home. It's an antacid. It also says pain relief. Now, they sell this actually not for science classrooms to add CO2 to water. They actually sell it because it is a pain relief and it reduces acid, like in your digestive system. So people who have heartburn, it's because you have acid inside of your esophagus. So this reduces the acid there, so you don't have that chest burning pain. It's also, though, helpful for pain relief. It says here it's a fast relief of acid indigestion, upset stomach, heartburn, headache, body aches, and pains. So there's also aspirin inside of these pills as well, or inside of these tablets. But we don't need the aspirin part. What we need inside of these is the parts of it that allow us to create CO2. So let me show you the chemical reaction that happens when we put one of these in the water. There's actually something else that's in here that's we call vitamin C. Vitamin C is also known as citric acid. So we have citric acid and baking soda is what makes up an Alka-Seltzer tablet. When you put it into water, it ends up creating, well, more water, sodium citrate, which is a type of salt, and then also CO2. The only thing that we're going to be focused on that we need from this is the CO2. So we'll get some extra stuff too, but this will not affect our experiment. All we need is the CO2 from these tablets. So the reason then that we're using Alka-Seltzer is to add our CO2 into our liquid. Now we have CO2 in our breath, but you can't just blow your breath into the water and cause it to have CO2 in it. The seltzer part of Alka-Seltzer, think about seltzer water. It's all fizzy because it has CO2 in it. So the reason they call it Alka-Seltzer is because we're going to be getting the CO2 out of this process. So you'll see a bunch of fizziness in this beaker here. Okay. Now the problem with this is that if you have a bunch of fizziness, it can actually cause this to be a problem. So right before you put this one underneath the lamp, if you could come talk to me, I'm going to show you a little trick of something we can do to make this one better. So before you put it in the lamp right here, if you're in group A or whatever group adds the CO2, then I want you to come see Nickel. Okay, come see me. All right, Another thing I'm going to ask you guys to do is add a little tiny bit of soap to each of these. Now, we're going to be using soap a lot in this unit, actually. You're going to see that when we do elephant toothpaste, you're going to have to add soap. And when we're doing the leaf discs, I'm asking you to add soap. The soap is going to play a different role each time, but it's never actually part of the reaction. Soap is there for many different reasons, and it helps us in chemical reaction to be able to, well, we can use soap as like a tool. And I'll show you why it's a tool. So let's watch this video right here. It was actually made, well, it looks like it was made from a fellow middle schooler. It's just a really quick overview of why and how soap works.
How does soap work? A soap molecule has two ends. One end is hydrophilic. This end is attracted to water, and the other end is hydrophobic, which is attracted to oil. This is what makes soap so awesome. Oil and water don't mix. The hydrophobic side of soap attaches itself to oil and dirt particles. A bunch of different soap molecules attach themselves to these particles. So, with all these soap molecules attached to this one dirt particle, when the faucet releases water and it hits these soap molecules, it pulls down on them and is pulling it off a person's hand, which is also pulling off particles of dirt and oil, which is in turn also pulling off germs. So now you know how soap works. Thanks for watching.
Okay, so in this experiment, we're using soap for that reason right there. This is what a molecule of soap would look like. It's just a fat, really. It's just a fat that has an acid on the end. So you have one end that wants to attach to the soil or to the something. It wants to attach to something that doesn't like water. This part likes water. So in one hand, it's grabbing a hold of the dirt, and then the other hand, it's like reaching for the water. And when it grabs the water that's flowing past it, it grabs the water and it flows with the water. So that's why we use soap to wash ourselves with, like our hands or our bodies, because if you have dirt on yourself, water by itself won't just get rid of the dirt and the oils that it's on our body that makes us dirty. You need the soap too, because the soap attaches to the oils, and then it helps pull those oils off of us. So if we look at what's happening inside of our leaf cells and why we're using soap, because if I draw a box here to symbolize a plant cell, here's like the nucleus and the endoplasmic reticulum and the central vacuole and a bunch of chloroplasts and stuff. If we look at the actual cell membrane in this region right here, if we could zoom in right there, we would find that there's all these little things that are, a bunch of these circles are the heads of these molecules that have these two legs on them. The two legs hate water. Like, they do not want to be in water at all. So what they do is, since they don't want to be in water, there's water on the inside and on the outside of the cell. So you have water here on the inside, and we have water on the outside of the cell. Now, these things hate water. So what they've done in cells is that there's actually another layer of them over here, because they're like, oh, I don't want to touch water. So they just have a double layer. These things are called phospholipids. But now these two legs are next to each other, so they don't have to touch the water. So this is why, if there wasn't a double layer here, then water could just easily go in and out of the cell. If it went in and out of the cell very easily, then the cell would actually just get bigger without even wanting to, because the water would automatically enter. So it would get so big that it would burst. If it didn't have this layer, then it would just lose water as soon as there was no more water around, and the cell would dry up. But since we have this layer here, water cannot enter or exit unless the cell wants it to. And the cell actually has these certain gates that open or close if it needs water. So it has little gates here that let water either enter or exit. Now what soap does for us is soap is like an Uber driver, and it's like, oh, what's up, water? You want to go inside the cell, do you? Well, I know how to get in there. I know the secret password. I can drive you in there. And so the Uber driver, aka soap, takes the water from the outside of the cell and transports it into the cell. So we need we need soap in both of our water solutions, because what we're going to do is we're going to force this water to enter the cells of our leaf. The way we're going to do that is we're going to create a vacuum using some syringes. So this syringe, you can take the plunger out of the syringe. You're going to put the leaf pieces inside of here at one point, and then you're going to put the plunger back on, and you're going to get it set up. Now, if you've ever seen the movies, you know, where there's like a doctor and he's about to stick a needle into somebody, and it's like, you know, they're looking at it and they go, and then water goes, like some water, like whatever that was in the syringe, like squirts out a little bit. The reason is because if they give you a shot and there's actually air inside of here, it could kill you because you get air inside of your veins, and that'd be really bad, like deadly bad. So they do that to make sure there's no air left inside of here. You're going to do that so that there's no air left inside of here. But then what we want to do is we'll just have the liquid. Either you'll have just water and CO2, or you'll have just water, or you'll have water and CO2 inside of your syringe. So in order to force that water inside of the cells, you're going to put your finger on here. And this is kind of hard to do. Last class, there was like people were doing this with 2 people. It was kind of hard to pull this back. But you can pull it back. And then what it does, it creates a vacuum inside of here. You can see it wants to go back up because it creates a lot of pressure. That pressure forces the water inside of our cells. If you pull too hard, then you might pull this out, and then your water and your leaf pieces come out of the back of the syringe. Now, it's really hard actually to pull the syringe out of this big one. If I have these little ones over there too, it's really easy to pull the back out of those. So just be really careful. The bigger ones definitely have more pressure inside of them, so it's easier to use these. These will still work though. What our syringes do though is they force that water. The syringes help force the water and the CO2 into the cell, or just the water into the cell. And the soap is like the thing that allows that to enter the cell. So that's how we make a vacuum. Then you'll know your vacuum works when all of your leaf pieces sit at the bottom of your beakers. We'll turn on the sun. We'll record how many of these things float. What will make these things start to float? What's going to make our leaf pieces? If they're all sitting at the bottom, there's no air inside of them at all. There's no gases inside of them at all. If a gas is produced, then the gas will make that leaf piece float up. So if we turn on the sunlight to these leaf pieces, what would we expect to happen that might make them float? What do you think, Keegan? And why photosynthesis? If photosynthesis happens, and it's going to take our CO2 that's inside of this liquid, and it's going to turn it into oxygen. And oxygen is a gas, and so if a gas is produced inside the leaf piece, then it will float up because there's a gas in it. So what we're looking for is how many of these pieces float in each of these. So if you start with it floating, you're not going to be able to get any data that's going to be useful. So this is our overall experiment. We'll talk, and we can distill more information out of this experiment, but I think I've given you enough to at least run the experiment. Like I was saying, it does take a long time to get this experiment. There's a lot of steps here. So it's important that when you guys are working through these different steps, you're asking me if you need some help. All right, remote students, welcome to the experiment process here. So here there's a group that is using the spinach leaves that I provided, and they're using a hole punch to get out some leaf discs. They put them in that syringe. They added their water with CO2 or just the regular water. Inside of there, we had to make that vacuum, force that all inside of the plant cells. But you can see here, it's pretty tough with these massive syringes. Some people did have a smaller syringe though, which made it a little bit easier to make the vacuum. But they had to do this many more times here to really get it in there, to get the liquid to go inside the leaf discs. To begin with, we would know that the leaf discs didn't have any extra air in them if they were all starting there at the top. Now, here's a time lapse of one of the experiments. Once you have the 2 beakers under the lamp, you can see that one of the beakers there on the left, none of the leaf discs are rising to the top. On the right, there are leaf discs that are rising to the top. The beaker on the right has those little bubbles in it, and that means that that beaker had the CO2 in it. The one on the left did not have any CO2. So we can see that the beakers that have the water plus CO2 in them end up causing photosynthesis to happen, meaning that oxygen is produced by the leaf cells. When oxygen is produced, those leaf discs then rise up towards the surface. Here's another group's experiment as well. The one on the left this time has little bubbles in it. You can see the CO2. That means it has the CO2, and you can watch these little leaf discs rise to the top. The reason they're rising is because they're producing oxygen. The oxygen leaves the leaf, but it can't quite leave it, so it attaches to the little leaf piece, and it causes the leaf piece to rise. So this was our experiment. If you need to add data to your data table, you can see here that on the left, all 10 of the leaf discs rose. On the right, none of them rose. So this took about 15 minutes in order for this to actually occur. So in your data table, you can add 15 minutes and say that 10 out of 10 of the leaf discs rose in the CO2 beaker and none of them rose in the regular just water beaker. That's our carbon capture technology experiment.